undergo senescence or death, right? So here, the cell cycle. So the life of the cell is called a cell cycle. That means the reproduction of the cell. And this cell cycle consists of different phases like G1, S, G2, M. So G1, S, and G2 are called interphase. So which I have already discussed yesterday. And there is M phase also. So cells that have temporarily or which have stopped dividing, they enter into the quiescent state, that is G0 phase. I told you that nerve cells, mostly they enter into the G0 phase. So I'll just rewind if any of you missed yesterday's class. So the G1 phase is the preparatory phase for the S phase. So G1 is the gap 1 phase where the time taking for organelles like protein synthesis, everything occurs, and it will also prepare all the proteins, enzymes required for the DNA replication. So uncoiling of DNA, everything takes place. It is a preparatory phase for the S phase. And most cells that differentiate will go during this phase, and some cells will get arrested in the G1 and they will enter into the G0 phase, which they cannot reproduce further. Okay, so certain cells in the G0, when given some external or internal stimuli or like signals, they can revert back into the G1 phase. Okay, and they can continue the cell cycle. So like usually nerve and muscle cells. So you don't find the regeneration of the, once they get damaged, they are damaged, right? So we don't see a regeneration of the nerve cells and the muscle cells because they enter into the G0 phase. Then the next phase is S phase, which is synthesis phase, where the DNA replication takes place. So this cell cycle is seen in the eukaryotes. Yeah, sorry, ignored. So the cell cycle takes place in the eukaryotes. And here, the DNA replication actually takes place. Whereas in prokaryotes, the replication usually takes place just before the cell division. Okay. So in the S phase, the DNA uncoils. So there are different steps that take place. So DNA uncoils. So before DNA replication, the DNA has to unwind because, you know, in the replication, DNA polymerases will actually polymerize the DNA in the DNA replication. So for the access of this DNA polymerases, this DNA has to unwind. So I hope that you have a basic information about the DNA replication, right? So what are the enzymes which are involved in DNA replication? They are DNA polymerases. So this DNA, when DNA is double-stranded, there will be no access to the enzymes or any other molecules. But during replication, the DNA has to unwind itself so that it can allow the DNA polymerases to enter and to have an access to the template strand of the DNA so that it can carry out the replication. Then other organelle replication, like other organelles like mitochondria, all these have this DNA. So all these replications, everything takes place during this S phase. Then the G2 phase. So G2 phase of the cell cycle is again the gap 2 phase, which is a preparatory phase for the mitosis. Okay. So here, this occupies the time from the end of the S phase to M phase, that is mitosis. So here, once the DNA is replicated, so now it has to check, the proofreading has to take place. That means whether the DNA which is replicated is error-free or not. That is, it is correctly replicated or not. Sometimes what happens? There will be more errors. So it is the replication is not error free. Okay. There is always a chance of error. So whenever there is an error, there are two options. One is repair enzymes comes and they will repair the DNA. That is one option if the DNA is repairable. If the DNA is not repairable, then the cell will undergo cell death. Okay. So all these like checking of DNA, whether it is correctly replicated or not, all this takes place during G2 phase. And other necessary proteins like tubulin, microtubules to make spindle fibers, all these are prepared in this G2 phase. So this phase takes place for 4 hours. Okay. Then next important phase is that is cell division, that is M phase or mitosis. So the somatic cells, they usually have this mitosis, whereas the germ cells or reproductive cells, they will undergo cell division through a process called meiosis. Okay, so during mitosis, the nucleus is replicated and even the cytoplasm is also replicated, and all the organelles, everything gets replicated and they will separate it into 2 daughter cells. So as DNA is already replicated in the S phase, so that is utilized in the mitosis. So here, so what is that is allowing this mitosis? That means without any cell signal, this cell division won't start. Okay, without any signal. That means there will be some external signal which will be triggering the cell division. Okay. So here there are 3 checkpoints. Before entering into the mitosis or M phase, there are some checkpoints that the cell will undergo. Okay. So they are nothing but internal controls. So the control of the cell cycle is dependent on the accumulation of signal molecules. So here, these signal molecules has to phosphorylate to get functional. That means they will be inactive usually, but if they have to get activated, they have to be phosphorylated. So what are the signal molecules present here? They are cyclins and kinases. So cyclins are the proteins and kinases are the enzymes which will transfer the phosphate group to make them activate. So here these kinases are in turn dependent on the cyclins. Hence they are called cyclin dependent kinases. So cyclins are a family of proteins that control the progression of cells through the cell cycle. That means the cell will check at every stage, that is G1, S, G2 and M phase. So every phase should be proper. Only then the cell cycle or cell division will complete. If it is not correctly done, then it will stop there. Okay. So here, these are the proteins. Cyclin proteins are the actual ones which will monitor this cell cycle. So here they will activate this cyclin dependent kinases, which are the enzymes. And only with the cyclin, in the cyclin dependent kinase will be active, because the name itself you can see. Cyclin dependent kinases means the kinases which are dependent on the cyclins. So these cyclins were named after their concentration varies in the cyclical fashion during cell cycle. That means that means I told you the checkpoints we have G1 at G1 will have one checkpoint, S phase will have one checkpoint. So based on these checkpoints, there are different cyclins. So these cyclins concentration, as it vary across the cell cycle, they have got the name as cyclins. So then a kinase is an enzyme that transfer the phosphate groups from high energy donor, that is ATP, to the substrate. So here that means this proteins has to get activated by the addition of phosphates. So from where does this phosphate comes? The donor is always, mostly 99% will be ATP. And ATP will donate the phosphate group and it forms ADP and PI, inorganic phosphate. And this process is called as phosphorylation. So these cyclins, as I told you, they fluctuate throughout the cycle. That is, they have cyclin D, cyclin E, cyclin A, cyclin B, like that. So some cyclins have, okay? So I'm not explaining you in detail, okay? So these cyclin dependent kinases will actually regulate this cell cycle. So yeah. So now you understood that there are cyclins and cyclin dependent kinases. Now I will explain you the stages of the mitosis. Okay. I'm not explaining you in detail because it is not much necessary for you. Just remember that the regulation of cell cycle is it is regulated by various checkpoints like cyclins and cyclin dependent kinases, which are called CDKs. So this is about the cell division, how it happens. So most cell division results in the daughter cells. Okay, so only meiosis occurs in the we will get the haploid cells. Mostly this mitosis, it will give you the diploid cell. That means it will have two copies of the genetic material, whereas meiosis give you the haploid cells. That means it will have only one set of genetic material, whether it is sperm or egg cell. Now, so all these genome chromosomes that I have explained. So this is the original image of the chromosome, how it looks. So this is the first step where the chromosome will have, and the centromere is at the center, and the arms, chromosomal arms. We call it as long arm and short arm. That is called P arm and Q arm. We usually name. So when you are telling about any location of the gene, you usually tell that it has where it is located on the P arm or Q arm, and you will have some position. That is, suppose 6th chromosome P arm, and you have some 22.3 like that location. So you have to tell that this gene is located in 6P 22.3 like that. That means 6th chromosome P arm and what is the location. So this is a single chromosome. Here, when these two sister chromatids they join together, so that is the chromosomal duplication. So here is the real image you can see. So once this duplication takes place, these two chromosomes will segregate into the two different cells to form two daughter cells. So this eukaryotic cell division consists of two stages. One is mitosis and cytokinesis. So mitosis is nothing but the division of genetic material in the nucleus, and cytokinesis means it is the division of the cytoplasm. That means once this genetic material is replicated, so this genetic material is separated into the two different cells, and then the cell cytoplasm also will divide, and finally the cells will divide to daughter cells. Okay. Whereas meiosis, actually it yields into the two unidentical daughter cells. Okay. So the mitosis phase alternates with interphase in the cell cycle. So I told you what is interphase and mitosis. So in 1882, German anatomist Fleming, he developed some dyes, okay, to observe various chromosomes during mitosis and cytokinesis. That means during cell division, how they are dividing. So only after use of these dyes, he could identify so how the cell division is taking place. So this M phase, it consists of both mitosis and cytokinesis, right? So I'm not explaining you again. So this is how it represents. So the top one is the original image and bottom one is the image C, okay? So in the G2 phase, I will show you. So in the interphase, that is G2 phase, which is before M phase. So how it looks like is, so all the genetic material is divided, now ready to segregate. So you have nuclear envelope, nucleolus, everything, and centrioles with two centriole poles are already synthesized and the chromosomes are duplicated and condensed. Then the first stage of mitosis is prophase. So in the prophase, you can see the top one is the real image, so which you cannot understand, but for your understanding, you can see for your observation. So early mitotic spindles are formed and they are attached to the asters. So towards the poles. And now the two sister chromatids of each chromosome are formed. And in the pro-metaphase, these fragments are attached to the, the spindle fibers attached to the kinetochores. And here these microtubules will pull these chromosomes towards the poles. So in the metaphase, actually they are all aligned into the metaphase plate that is at the center. Now, centrosome is located at each pole. Okay. Now in the anaphase, now these chromosomes are pulled towards each poles. And in the telophase and cytokinesis, these chromosomes, they are separated into the two cells. A cleavage furrow is formed. Cleavage furrow is nothing but like a bending to, it is like an imaginary line where the division has to take place. And nucleolus is formed. So the same thing, yeah. So this is how it looks like. So what is this mitotic spindle? So it is nothing but microtubules, I told you, which is especially important for the movement. So in animal cells, these spindle microtubules begins in the centromere, where the microtubules are organized. And these centrosome, sorry, not centromere, centrosome. So this centrosome replicates during the interphase, that is at the G2 phase, forming two centrosomes that migrates to the opposite ends during prophase and pro-metaphase. And here an aster, I told you in the image. So the aster, this extends from each centrosome. And this spindle fibers, they include microtubules, centrosomes, and asters. So these together are called spindles. During pro-metaphase, some spindle microtubules attach to the kinetochore of the chromosome and they begin to move to the center, right? Okay, they will align at the center. So these kinetochores are the protein complexes associated with the centromeres. Now, these chromosomes are all lined up at the center, which is called as metaphase plate. That is a plane which is midway between the two poles. That is at the center. You can see here. So even this is the real image. This is the real image and this is the imaginary, like graphic for your understanding. In the anaphase, the cohesins are again another proteins. They are cleaved by the enzyme which are called separase. Now, until now, these two sister chromatids are attached to each other by cohesins. But these cohesins are cleaved by the separase. So now these two sister chromatids, they separate and move along the kinetochore microtubules towards the opposite ends. Now these microtubules, they will shorten. So here you have a concept that is called polarization and depolarization. So these microtubules, they usually polarize, means they will move. The length will be more. That is called polarization. So whenever the chromosomes has to pull towards end, the length of the microtubule has to decrease. That is called as depolarization. Okay, that happens at their kinetochore ends. So there are some non-kinetochore microtubules from poles which will overlap and push each other along in the cell. So in the telophase, that is the last phase, so genetically identical daughter nuclei are formed at the opposite end of the cell. And during cytokinesis, after this telophase, the spindles usually disassemble. So in the cytokinesis, in animal cells, this cytokinesis occurs through a process called cleavage. First, it will form a cleavage furrow. But in plant cells, a cell plate is formed. So that is the main difference between the animal cell and the plant cell. So here you can see, this is the animal cell where the furrow, that is a deep bending is seen here. Okay. So a contractile ring of microfilament is formed in between. Whereas in plant cells, you can see a cell plate. So this is the animal cell one is SEM image. This is scanning electron microscope. And here in the plant cell, this is STEM image, then transmission electron microscope image. So these are the original images of the cells. Okay, like this is the prophase, how it looks like. Then pro-metaphase, metaphase, then anaphase and telophase, where actually the cells will the two genetic material is separated into two daughter cells and cytokinesis takes. So whereas in bacteria, if you see, the cell division is mainly by binary fission. So not only in bacteria, even in archaea, that means in prokaryotes. When the chromosome replicates from the origin of replication, which is located in the bacteria, so from where actually the replication starts. And the division of, replication of DNA also happens just before the cell division. So here the two daughter chromosomes move apart. So here you don't find any chromosomes as such. So mostly it will be segregated form of the DNA. So circular DNA like that. Okay. And the plasma membrane pinches inward and dividing it into two. That is called binary fission. So here how it looks like, you have a green color one. Can you see? That is the origin of replication. And the cell will have a cell wall, then plasma membrane, and you have, this is the chromosome. This first, from the origin of replication, this DNA replication takes place. And here two copies are formed. And now you can see two origins of replication. Then the replication finishes when this that is a division or a what you call separation is seen, and it will divide it into two daughter cells. That is called binary fission. So how this mitosis has evolved? So already we know that evolution takes place from the prokaryotes. So you can see only this this mitosis in the eukaryotes. So how this evolution of mitosis has taken place? So since prokaryotes evolved before eukaryotes, so mitosis might be probably evolved from the binary fission. That is the theory. So certain protists exhibit this kind of cell division where it is intermediate between the binary fission and the mitosis. So here the eukaryotic cell cycle is regulated by the control system. Okay, that is a regulation at the molecular level. So what is the cell cycle control system means? It has some chemical signals that are present in the cytoplasm. So that will actually control the cell cycle. So they are called as checkpoint, as I told you. So there are some internal controls and external controls. So this is how the checkpoints looks like. Okay. So you have G1M, G2, G1S, G2M phase. So here you have at the G1 one checkpoint, and after G2 you have one more checkpoint, and after M phase you have one more checkpoint. So the cell cycle clock includes cyclins and cyclin dependent kinases, as I already explained. But again, I will just brief you out. So there are two types of regulatory proteins, cyclins and cyclin dependent kinases. So the activity of cyclin dependent kinases, it rises and falls as the concentration of its cyclin partner changes. That means if the cyclin levels are changed, concentration is changed, then cyclin dependent kinases concentration also will change. Okay. And here you have maturation promoting factor, which is a cyclin CDK complex that triggers the cells pass the G2 checkpoint into the M phase. So after G1 checkpoint, if the cell enters the S phase, once the DNA replication takes place and the produced daughter DNA is correct, now this from synthetic phase, it enters into the G2 phase. So after G2 phase, if everything was properly done, then it enters into the M phase where you have maturation promoting factor, which is involved. So here many signals, they are responsible for this cellular surveillance mechanism. So that means various signals that will be keeping surveillance, monitoring whether the cell cycle has correctly done or not. So there are three important checkpoints, that is G1, G2 and M phases. G1 checkpoint seems to be most important because if a cell receives go-ahead signal at the G1 checkpoint, then it usually completes the S phase, G2 and M phase and divides. That means after G1 checkpoint, if everything is correct at G1, then there will be a go-ahead signal. That means, the cells now can pass the next phase, that is S, G2 and M phases. And if at G1 checkpoint, there is okay signal, then the other three phases will be done successfully. If the cells does not receive this go-ahead signal at G1, then the cell has to exit from the cell cycle and it has to enter into the G0 phase, that is non-dividing phase. So here you can see, this is the G1 checkpoint. So this is how it has to move. You have the red, this is just for symbolic. So without full chromosome attachment, if there is no full attachment, then this is for M1, M2, G1. Okay. So then what happens in the first phase, this G1 has to go into the S phase, that is replication phase. So here the DNA has to unwind properly and all the proteins, enzymes required for the DNA replication has to be synthesized and they should be properly arranged. So if anything is not correct, then it will enter into the G0 phase, revert back into the G0 phase. And if everything is correct, then it will pass the G1 checkpoint and enters into the S phase. So here, once everything is correct, then it will get the go-ahead signal to continue the cell cycle. So, another one is, okay. So in the G2, what happens? It comes after S phase. So S phase where the DNA replication takes place. So full chromosome attachment, everything has to happen and there are no errors in the DNA replication. Everything is correct. Then G2 checkpoint will say, okay, you can go ahead. Then M phase, that is M checkpoint. So if there is no proper attachment, then it will go back, or the cell will undergo death. Or if everything is correct, that is even after M phase, that is the replication has happened, the chromosomes have moved. So what happens, the main error that happens after M phase is that if the chromosomes, the two copies has to move to the poles correctly. If both the copies have moved to only one side, like that, if any error happens, then the cell cannot enter into the this phase. Okay, the next cell division cannot be completed. So what happens now? The cell has to undergo death in such case. So then internal signal that cell will not begin anaphase until all chromosomes are properly attached at the spindle at the metaphase plate. So this mechanism assures that daughter cells have correct number of chromosomes. That means each that means during anaphase, like all these chromosomes has to correctly divide into in between two daughter cells. If both copies goes to one daughter cell, then the other daughter cells, the chromosome will be absent. So that can cause various genetic disorders. So mostly here the cell will undergo death. But some cases, so now we can see like various genetic disorders where we see this chromosomal abnormalities, like having an extra chromosome or absence of one chromosome or one copy of chromosome. They will give rise to different kinds of diseases, like you can see Down syndrome, all this, right? So the external factors that regulate the cell division include growth factors. So there are various growth factors which are released by the cells, and these growth factors will regulate the other cells to divide. They will give some stimuli. So some of the growth factors examples are platelet derived growth factors, which is mainly made by the blood cell fragments, that is platelets. So platelet derived growth factors are produced by the platelets. And here the main purpose is, so another important function here is density dependent inhibition. That means these are the external signals. Okay, so one external signal is growth factors. Like when the adjacent cells, they produce some growth factors, they will induce the division of the other cells. That is one kind of external signal. And in another external signal is density dependent inhibition. That is, if the cells are crowded, then what happens? Usually the cell division will stop. So that is like a death signal, not death signal, but it is an inhibitory signal. But in some cases, this kind of density dependent inhibition is absent. So in such cases, we can see some diseases. So this is like how this density dependent signal looks like. So whenever we take a tissue of human and if you keep on a Petri plate where you will pour agar in the medium, not agar exactly, you will get animal cell culture plates will be different. So you will have matrix on that. So and that you have to add the media and the tissue piece. Then when you culture it, and if you add platelet derived growth factor to that, so you can see how it looks like. So this in cell plate without platelet derived growth factor, the growth of the fibroblast is very less. Whereas in the presence of PDGF, you can see an increased growth. Okay, more growth is seen. So this is the same image of the culture fibroblasts, how they look. Now, so most of the normal cells, they exhibit anchorage dependence to divide. That means the cells has to attach to something that is substratum so that it can divide. Okay. So this density dependent inhibition and anchorage dependence, these are very important factors for the normal cells. If any of these two are lost, these are like symptoms where the cells will become the cancerous cells. Okay, so this is how it looks like. So here the first step where you can see the cells are attached to the bottom. So that means it is anchorage dependence. And when the division has to stop, if in cancer it is full, still they are growing. The cell division is not stopping. So here you can see the division between these two cells. So difference between these two cells. So this is a normal cell and this is the cancer cell. So how they are different. Okay. So usually loss of cell cycle control leads to the cancer cells. So how this cancer grow? Mainly by factors, and they convey a growth factor signal without the presence of growth factor or abnormal cell cycle control. That means they will pass these checkpoints, whatever they are present. And the normal cell will be transformed to the cancerous cell. Okay, some cells will become benign tumors and some will be malignant tumors. So benign tumors are nothing but the tumors which are mainly confined to a local area. That means they won't spread and non-dangerous mostly. Malignant tumors means they will spread. So they will metastasize. That means they will pass to the another place where other than the origin of the cancer cell, and they will spread. So they are dangerous. So here, so I'm not going into this cancer topic. So mainly like after, so the purpose of understanding the cell cycle and cell division and the checkpoints is mainly now the research is advanced towards the cell cycle and cell cycle signaling in cancer treatment. So now we have to like personalized medicine is the another field of research. So now that means we should know where exactly the problem is, and accordingly we can treat that particular tumor, because the same treatment is not applicable for all the tumors and all the human beings. Okay. So this is all about the cell division. So if you have any questions or if you don't understand any concept, you can ask me. I will explain you again.
Ma'am, it comes under mitosis only.
Which one? Yeah, whatever I have explained you is mitosis only. No, I am not explaining, like cancer is, I don't know whether it is there in your syllabus or not. Okay. Means why I am telling you why it is important. Why you are like studying the mitosis is not just for, means what happens if the control is lost. So then that leads to the diseases like cancer.
Okay, okay.
Is it clear?
Yes, ma'am.
Yeah. So that means simply just remember one thing, that all these concepts like cell biology and mitosis or whatever it is, so they are introduced from our schooling. Like we are studying from like mitosis, cell division, everything. I hope in school also we might have studied. So all these phases, prophase, metaphase, anaphase.
Yes, ma'am.
Now, yeah, but
We
in
forgot.
that, yeah, sorry.
We forgot also, ma'am.
Yes, yes. Yeah, I can understand. So, but now how you have to think is in school days, it is only for our understanding, right? It is a basic, okay, the cell will divide in these steps. But now as you means coming to the upper classes, now you are UG. Then and PG. Then after, like, why we are studying this? Already scientists know that why mitosis is happening. So what are the phases? Then what is the use that we are studying? That means how we can apply this knowledge in the research. So it is ultimately, as a science students, we have to think like how we can actually make them come into the real, like how it can be helpful in the real world, right? So that's why now we know that mitosis happens, all these are the steps. Now what happens if the steps are failed? And what are the symptoms? See, in the cell, the steps will fail or it will be successful. We don't know. But ultimately, whatever the change that is happening inside the cell, so it will come out as a symptom or a phenotype, right? So when it comes as a symptom, then only we know that it has become a disease. Once we know the disease, once we know, like suppose cancer, now a lot of research is going on the cancer. So once we know, okay, the cancer, it is diagnosed, then comes the treatment. So why all the drugs are not working, right? So though there is lot of research, lot of drugs have been identified, then why it is not working? Because the underlying cause is not known. Once we know what gene is mutated, at which stage it is mutated, all these things we know, then we can actually design a proper drug. And that too, each individual have different background of developing a disease. So that is called as personalized medicine. So what I suggest you is not only just for exam point of view, but you can also explore. So what is the current research which is going on, so that you can better understand the concept. Okay. So do you have any more questions regarding today's class? Any concepts?
Ma'am, can you just briefly tell prophase, pro-metaphase and metaphase and telophase?
Yeah. So initially, what happens after interphase? So I will show you the image so you can better understand. Yeah. So here, this you understood. After S phase, in the S phase, DNA is replicated, but it is not made it into chromosome. Okay, only the DNA is replicated. Now you have the 2 copies of DNA which are scattered around the nucleus. So now you can see how the nucleus is looking messy. Okay, lot of network of DNA, and you have centrosomes at the top, yellow color ones. So they are with centriole pairs. Okay, they are synthesized. So this, now the cell is ready for the division. Now in the prophase, what happens? This DNA, which is replicated, now that is organized in the form of chromatids first. These chromatids are attached at the centromere to form sister chromatids. Now these are the chromosomes. Now the chromosomes are ready in the nucleus. And here from the centrosomes, here you can see, now microtubules, they form spindles. So you can see from both the ends, a string-like structures are formed. They are called spindles. In the pro-metaphase, so in the metaphase, before just metaphase, there is called pro-metaphase. So here what happens, these spindles, actually they will attach to the kinetochores from both the ends. That means these centrosomes, they move towards both the ends, that is poles, and from both the ends, the spindles will come and they will attach to the kinetochores of the sister chromatids of each chromosome. Okay. And there are some non-kinetochore microtubules which are not attached to the chromosomes. That is kinetochores of the chromosomes. They are separated. Now, this nuclear envelope is fragmented because a new one has to form. Now, what happens once the attachment of spindles to this kinetochores happens, all the chromosomes are aligned at the center, which is called metaphase plate, and this stage is called as metaphase. Okay. Then in the anaphase, these two sister chromatids, which are attached by cohesin at the center. So the cohesin will be digested and these two sister chromatids are actually separated and they will move to the pole. Once they move, now the center will form a furrow, that is cleavage furrow, where actually the division has to take place. Now you can see that these two sister chromatids are towards the opposite side, and at the center you can easily see the division. So here actually cytokinesis, that means the cytoplasm also will get divided into both the cells, and now finally this nucleolus is formed and nuclear envelope is formed and everything happens, and this cell division takes place at the center. Now the two daughter cells will be formed. Okay. Is it clear?
Yes, ma'am.
Okay. So if there are no questions, I will stop this class today. Right?
Ma'am,
Okay.
can you share the PPT in the group?
So am I there in the group?
I don't know, ma'am. I have to see. Okay.
Okay.
You give your number, I will add.
I will just give you in the chat. You can add me. I will.
Okay.
Okay. Yeah, you can add me so that I will share the PPTs. Okay. Okay, fine. Thank you so much.
Okay, ma'am. Thank you.
Yeah. Thank you.